Hello students, welcome back to your own channel, that is Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. Students, before we start today's amazing class, quickly let me know in the chat if I am audible to all of you. That is sound check, and the presentation behind me is visible to each and every one of you. Like it is perfectly visible and I'm perfectly audible to all of you. And if I am audible and visible, quickly mark your attendance. That is, all the students who are watching it live. It could be your Ravindrani, Harbeyam, Virat, Sebastian, Nala. All of you, quickly mark your attendance. And also, if you're watching it recorded later on, also mark your attendance, because this is going to be a really, eye-opening session for you. Because we'll be doing mitosis. The title says we'll be doing mitosis. But in mitosis, students, I'll be teaching you every single line of NCERT. I'll be making sure you write down the notes. We will watch animations. That's right. We will visualize how the mitosis is happening, how the movement of chromosomes is happening, everything with the help of animations. And also, at the end of the video, we'll be solving a lot of questions. So this is going to be a complete package of your mitosis. And after today's session, after this video, you do not have to ever read mitosis. Obviously, you have to revise. Revision is there, but the concept will get clear to you. Now, what is this 2 and 2C, 4 and 4C? All of those concepts will be clear to all of you. Now, quickly green us in the chat if you are excited for today's class, because this is going to be really eye-opening class. I'm telling you, honestly, by the end of the session, you'll be telling me, thank you, sir. Okay. Now, let's understand mitosis with a most simplest thing. What is mitosis? If someone asks you, sir, what is mitosis? Can you tell me in a simple manner? Mitosis is nothing but the way cells reproduce. Yes, mitosis is nothing but a way a cell reproduces. Okay, cell reproduction, I can call it cell reproduction. But let's go back in time. To something called as cell theory. Quickly tell me in the chat who gave the cell theory. All of you will tell me the two main scientists who gave cell theory was what? Scheiden and Swan gave you cell theory. All of you will be like, yes, sir. Scheiden and Swan told that every single organism, it could be plants or animals, everything is made up of cell and a product of cell. Okay. But there came one more uncle. He's like, wait a minute. You are not able to explain how the cells are formed. That was Rudolf Virchow. Rudolf Virchow came and told that cells came into existence from pre-existing cells. Cells came into existence from pre-existing cells. That means a new cell is formed. A new cell is formed from division of a older cell. So we have an old cell, it is dividing and we get a new cell. That is technically mitosis. That is one parent cell. We have one parent cell which is dividing and it is producing two daughter cells. Now what is interesting about this? What is so interesting about this, students? One parent cell is dividing and giving rise to two daughter cells. The interesting thing part here is that both parent cell and daughter cell, both parent cell and daughter cells are identical. Both parent cell and daughter cells are same to same, or we can say identical. They're identical with each other. They're same to same. They say, right, same but same it is. It is same but same. But about mitosis, there is one more thing you should understand, that even though the cell is dividing from parent to the offspring, the number of chromosomes will remain same. That is, there is no change in number of chromosomes. If 46 chromosomes in the parent, there will be 46 chromosomes here. 46 chromosomes here, 46 chromosomes here. This was 2n. The offspring will also be 2n. Parent cell is 2n, daughter cell will be 2n. Now, tell you, can I call it equal? This is equal, right? So can I call your mitosis? Can I call mitosis as reduction equational division? Can I call it equational division? Because the chromosome number is same. The chromosome number is going to remain the same. So can I call it equational division? That is, the chromosome number is going to remain the same. So is my voice breaking still a lot? Tell me in the chat if my voice breaking still or is it gone? Is it proper now? Is it proper? Sound check, sound check. Abhi, Ravindrani, is it proper? Is it voice still breaking? Is it still breaking? Sound check, sound check. Tell me in the chat now quickly. Is it still breaking? I believe it's not breaking anymore, right? It is not breaking anymore. So what did we learn till now? We learned that cells reproduction is mitosis. We have one parent cell, it is giving rise to two daughter cells. Both parent cell and the daughter cells are identical to each other. The number of chromosomes will remain the same. That is, if the parent cell has 46 chromosomes, the daughter cell will also have 46 chromosomes. 2n to 2n, the ploidy is maintained, and since it is equal in number, I am calling it equational division. I am calling it equational division. Any doubt here? Ask me quickly. I believe there should be no doubt here. No doubt here. Right. Now, I can ask you, this is your mitosis. But where do you find mitosis? What cells are reproducing? Cells are reproducing. But how is it beneficial for us? Right? How is it beneficial for us if cells are reproducing? Answer is very simple. Now let's take an example. All of you, in some stage in your life, you were a single cell. Tell me, in some stage in your life, you were just a single cell. That was your zygote. All of you were zygote at some stage in your life, obviously, the starting stage of your life. Imagine if there was no mitosis. If there was no mitosis, there would have been no you right now. That is, from a zygote to a multicellular structure, that is because of mitosis. That is because of mitosis. All the way from the zygote to become a multicellular structure, that is because of mitosis. Or one more thing, simple. We tell plants keep on growing. We tell plants have something called as indeterminate growth. Unlimited growth plants have. How? What is the magic there? The magic there is very simple. Plants have something called as meristematic tissue. Plants have something called as meristematic tissue. Right? Meristematic tissue. What is the magic happening there? Inside the meristematic tissue, there is mitosis happening. Because of mitosis, plants are able to continuously grow throughout their lifetime. That is the magic. That is the magic there when it comes to plants. Okay. Now, that was your introduction to mitosis. But the main question is, where does the mitosis takes place? In the cell cycle, where does the mitosis takes place? Students, if you know, this is your entire cell cycle. The entire cell cycle can be divided into two parts. The first part is called as what? The first part is called as your interphase. It's called as interphase. The other phase is called as what? M phase. Interphase as well as your M phase. Right. Interphase as well as M phase. Now, can you see this light blue color? This light blue color, the entire thing is what? The entire thing is your, this light blue color, entire thing is your interphase. Entire thing is interphase. This small thing, can you see this small thing? This right here is your mitosis or M phase. Furthermore, interphase can be divided into three different categories. That is your G1 phase, S phase and G2 phase. Now we are not interested in this. Our main focus is on M phase. Our main focus is on M phase right now. Our main focus is on M phase. Clear? So in your cell cycle, the entire thing is called as interphase, where we have G1, S and G2. The small portion, do you see the small portion? The small portion is called as your M phase. M phase. That is what we are interested in. That is what we are interested in. And this M phase can be further classified. This M phase can be further classified into two categories again. The M phase can be classified into cheirokinesis and cytokinesis. Cheirokinesis and cytokinesis. The M phase. The M phase. The M phase. Cheirokinesis and cytokinesis. Now, what is the meaning of chiro? The meaning of chiro means nucleus. Nucleus. So, cheirokinesis was division of nucleus. Cytokinesis is what? Cyto means cytoplasm. Kinesis means division. So, this is division of cytoplasm. This is division of cytoplasm. Okay, sir. This is the division of cytoplasm. Cytokinesis. Now, cheirokinesis is divided into 4 stages. How many stages? 4 stages. How do you remember this? Simple example. PMAT. It's called as PMAT. That is P-M-A-T. What is P? P is for prophase, prophase, metaphase, metaphase. Then we have anaphase, anaphase, and finally T for telophase. Prophase, metaphase, anaphase, telophase. Students, the order is extremely important. The order is going to be extremely important because many questions will come just from the order. They'll ask you a question, arrange the following stages of cell cycle. Arrange the following stages of your M phase in a particular order, in an ascending order, descending order. You should know it is going to be prophase, metaphase, anaphase and telophase. If you do not have any doubt till now, quickly green house in the chat because now we will be starting with prophase now. Can we start with prophase, students? Can we start with the prophase? One by one, I will show you the diagram. I will show you the animations so that you can understand what exactly happens in each stage of your mitosis. Okay? Clear? Now, can we start with mitosis? Can we start with your prophase now? Prophase. Now, what exactly happens in prophase? What exactly happens in prophase? Listen to me very carefully. Listen to me carefully. First of all, prophase is actually the longest phase. It is actually the longest phase. The main thing which is happening here is initiation, starting. How do you identify? There is initiation of condensation of chromosomal material. There is initiation of chromosomal material is happening. Initiation of condensation of chromosomal material. So chromosomal material is getting condensed here. Chromosomal material is getting condensed here. And students, your entire prophase can be divided into 2 categories. Into 2 different categories. One is called as your early prophase. Early prophase. The other one is called as your late prophase. Late prophase. Early prophase as well as your late prophase. Early prophase, late prophase. Okay? 2 different things. Now, what is happening in your early prophase? That is very important. That is very important. Now, this is your cell. This is right here is your cell. In the cell, what happens is, in early prophase, inside the cell there is condensation of chromosomes. Condensation of chromosome is happening. That is, this right here is your nucleus. This right here is a nucleus. Inside the nucleus, the chromosomes will start to condense. The chromosome will start to condense inside the nucleus. Inside the nucleus. Okay? Now, apart from that, what happens, students? Apart from condensation of chromosomes, there is something called as asters. There is something called as aster formation is happening. But you want to understand how is the asters formation is happening. You want to understand that. If you have learned the cell cycle chapter, cell the unit of life, there are two different structures like this, which are perpendicular to each other, called as centrioles, and together they are called as centrosomes. They are called as what? Centrosomes. So what is happening here exactly is, there is movement of centrosomes towards the opposite poles. Movement of what? Movement of centrosomes. And the centrosome has a amorphous substance around it. The centrosomes, which is consisting of centrioles, which has a amorphous substance around it called as pericentriolar satellite or pericentriolar space or centrome. You can call it anything, kinoplasm or anything. So all of you should know that. Okay. So what is happening here? Movement of centrosomes. Movement of centrosomes to the opposite poles. Opposite poles. Any doubt here? Ask me quickly. First one is condensation of chromosomes. Second one is movement of centrosomes to the opposite poles. So centrosomes are moving towards the opposite poles. Done. Any doubt here? Ask me quickly. Any doubt here? Ask me quickly. Next point is very important. Next point is going to be very important. That is, from this centrosome, students, listen to me very carefully now. Listen to me very, very carefully. From the centrosomes, there is microtubules are arising. From this centrosome right here, microtubules are arising. So there are two types of microtubules here. There are two types of microtubules. Yes. One which is arising here. Can you see? On the bottom, which does most of the anchorage and everything. Anchorage holds the centrosome at place and everything. The other one, other type of microtubules is what? Spindle fibers. Is the spindle fibers from the pericentriolar space. Spindle fibers are coming. Spindle fibers are arising. Spindle fibers are arising. So what is happening here, students? There is release of two types of microtubules. There is release of two types of microtubules, right? Two types of microtubules. Here. Two types of microtubules. Two types of microtubules. One is called as asters. The other one is called as your spindle fiber. Spindle fibers. Okay. The other one is called as spindle fibers. Okay, spindle fibers. Now, together, that is spindle fiber plus aster. Spindle fiber plus aster, they together they are called as mitotic apparatus. Mitotic apparatus. They'll ask you this question. They'll ask you this question. What is mitotic apparatus? They will ask you. They will ask you this question. What is mitotic apparatus? You should be able to tell mitotic apparatus is nothing but asters plus spindle fiber is mitotic apparatus. Clear? Everyone tell me in the chat right now. First, what we learn? In your early prophase, what is happening? Condensation of chromosomes. Chromosomes are getting condensed in the nucleus. Movement of centrosomes to the opposite poles. The centrosomes are moving towards the opposite poles. Now, from the centrosome, there are microtubules are rising. From the centrosome, there is microtubules are arising. Two types of microtubules. One is called as your asters. Other one is called as your spindle fiber. Other one is called as spindle fiber. Asters plus spindle fibers. Asters plus spindle fiber. It's called as mitotic apparatus. Mitotic apparatus. Everything is happening in your early prophase. Now, what is happening in late prophase? Students, sometimes what happens, you cannot distinguish between early and late prophase, so they're interchangeable most of the times. Okay. In late prophase, there is disappearing of Golgi complex, nuclear envelope, right? Nucleolus, mitochondria, everything is disappearing. Everything is disappearing. Clear? Disappearing of Golgi complex, nucleolus, endoplasmic reticulum, even endoplasmic reticulum, everything is disappearing. Everything is disappearing in late prophase. Clear? Yes or no? Tell me that quickly. Students, if you're able to understand this, I want everyone to quickly smash that like button. Your prophase is actually done. See this? What is happening in prophase? In your prophase, chromosomes are getting aligned here. Can you see chromosomes are getting condensed? Asters are moving. Can you see asters are moving to the opposite pole? This is your centrosome. Centrosome are moving towards the opposite poles. Moving towards the opposite poles. Spindle fibers are also arising. Also there is formation of asters. This is your entire prophase. Detailed structure. And students, you should know one more thing. How does it look like? When the compaction is happening, how does it look like? It looks like a ball of wool, right? This stage right here is also called as ball of wool stage. Or spreen stage. That is not in your syllabus, but if you can write down, Done. Notes. Are you making notes? These are the notes which you need to make. Which will help you in examination. Okay. Now, prophase is done. Right? Prophase is done. Movement of centrosome towards the opposite poles. Condensation of chromosomes. Now, this is your early prophase. Can you see nuclear envelope is there? Chromosomes are chromatin is just floating. But as soon as we come to late prophase, all the nuclear envelope is disappeared. Condensation of chromosome is happening. Okay. Now, next one is metaphase. What happens in metaphase, students? That is very important. Now, listen to me very carefully. This is your cell right now. Yes, this is your cell right now. In metaphase, now here we have, what is this? We have your centrosomes in the corner. Centrosomes are there in the corner. Okay. Now, in the case of your metaphase, all the chromosomes will align on the equatorial plate. Here like this. Can you see? All the chromosomes will align on the equatorial plate. And chromosomes are held together in the center. In the center, chromosomes are held together by a centromere. Okay. Now what do we have? We have the spindle fibers. Nice. Spindle fibers are there. Now I want you to write down few things here. Some things here. All the chromosomes are moving towards the center. That is called as movement of chromosome. The movement of chromosome is called as congregation. Okay. Important point. Congregation. Okay. The next thing I want you to understand is all the chromosomes are aligning. At a plate here. Can you see plate? This middle part. The middle part is called as metaphasic plate. One second, students. Metaphasic plate. It can also be called as equatorial plate. So all the chromosomes are aligned on the metaphasic plate or equatorial plate. Now, I want you to understand the structure of chromosome here. That is very important to understand. Structure of chromosome. How is the chromosome here actually? In the middle part, we have the centromere. Okay, middle part is centromere. This is a chromatids. These are the chromatids. Chromatids. Okay. They are called as your sister chromatid. Sister chromatids. Sister chromatids. One chromatid, another chromatid. The center part is called a centromere. Centromere. Now students, around the centromere, there is a protein layer, a disc-shaped protein layer. That is very important to understand. Around the centromere, here like this, there is a proteinaceous disc-shaped structure. Proteinaceous disc-shaped structure called as kinetochore. It's called as kinetochore. Kinetochore. Understand? Kinetochore. Now, next point is a lot of PYQs. Next point which I'm going to tell you. The next point which I'm going to tell you has a lot of PYQs. I told you the spindle fibers are attaching to the chromosomes. Spindle fibers are attaching to the chromosomes, I told you. But what is the point of attachment? What is the point of attachment? The point of attachment is nothing but your kinetochore. So all the spindle fibers, students, all the spindle fibers will attach to the kinetochore. All the spindle fibers will attach to the kinetochore, not to the centromere. Not to the centromere. They will attach to the kinetochore. Very important line. Many previous year questions, many PYQs. To open the book of a PYQ, this question will be there every consecutive year, or alternate years. This question will be there from this topic, kinetochore. So spindle fibers, these are your spindle fibers. These are your spindle fibers. So all the spindle fibers are attached to the kinetochore. Now you tell me in the chat, every chromosome has two kinetochores. One chromosome has two kinetochores. Two kinetochores. One chromosome has two kinetochores. In a diploid cell, how many kinetochores will be present? Tell me in the chat now. How many kinetochores will be present in a diploid cell? Quickly in the chat. Tell me. In a diploid cell which we have, where we have 46 chromosomes, we'll have 92 kinetochores. 92 kinetochores. Okay? 92 kinetochores. Remember that, because every single chromosome has two kinetochores. So 46 chromosomes, double of it will be kinetochores. Understanding? Every chromosome, students, every chromosome. One chromosome has two kinetochores. One chromosome, this is entire chromosome, this is one entire chromosome. They are chromatids. They are two chromatids. One chromosome has two kinetochores. So 46 chromosomes will have 92 chromatids, 92 kinetochores. Did all of you understand? Twice the number of kinetochores. That is very important to understand. And one more thing you should notice here. Is additional information. To one chromosomes, two spindle fibers attaching. To one chromosome, two spindle fibers attaching. That will change in meiosis. In meiosis, one chromosome, only one spindle fiber. But in mitosis, one chromosome will have two spindle fibers attaching. Two spindle fibers attaching. Clear, sweetie? Clear, Divya? Clear, Ravindrani? Jai? Mukunda? Divya? All of you understand the concept. Concept is very important, because all of this concept will make you understand even meiosis in a much more better manner. Okay. Now, the second point I want all of you to write down here is, second point is going to be spindle fibers. Spindle fibers attach to the kinetochore. Done. The third point about metaphase. Is that this is the best stage to study morphology. Best stage to study morphology of chromosomes. Morphology of chromosomes. Best stage to study. Best stage to study morphology of chromosomes. Done? Clear? All of you. Now students, this is your metaphase. All the chromosomes are aligning on the equatorial plate, also called as what? Metaphasic plate. Equatorial plate or metaphasic plate. Done? Understanding? 3 points done about your metaphase. The next one is gonna, this is just transition to the metaphase. All the chromosomes are aligning, but at the end of metaphase we have all the chromosomes with the 2 chromatids are attached at the equatorial plate. All the spindle fibers are attaching to the kinetochore. Can you see? If you open your NCERT, you'll be able to see all the spindle fiber are attaching to the kinetochore. Done. Okay. One second. And now you might ask, sir, why is it the best stage to study chromosomes? Simple. Why is it the best stage to study the chromosomes? Because at this stage, complete condensation has happened of chromosomes. Complete condensation of chromosomes has been done. Complete condensation is happening. Because complete condensation is happening, that's why morphology of chromosomes can be studied very easily. Okay? Done? Okay? See? That's why it's the best stage to study the chromosomes. Because complete condensation of chromosome has happened here. Okay? Done. Condensation is completed here. Now what is happening in anaphase? What is happening in your anaphase? Now students, here I want everyone to pay extra attention now. I want everyone to pay extra attention, because anaphase is the stage which has a hidden gem. It is a hidden gem. It has a hidden properties, and I'll be telling you that hidden properties. Okay? I want everyone to pay extra attention now. Extra attention. What is happening in your anaphase? What is happening in your anaphase, friends? Anaphase. Very important. This is your cell. Okay, this is your cell. In the corners, what do we have? In the corners, we have the centrosome. We have the centrosome here. Centrosome. From the centrosome, we have the spindle fibers. One second. We have the spindle fibers. What is happening in anaphase, students? In the case of your anaphase, all the chromosomes are splitting. Can you see? All the chromosomes are splitting and they are being pulled towards the opposite poles. That is, this was your chromosome till now. A chromosome with 2 sister chromatids. Now this is splitting in the middle. That is the middle part. What was the middle part, students? The centromere. The centromere was the middle part, right? The centromere will split now. The centromere will what happen? The centromere will split. If the centromere splits open, we end up getting, this is one chromosome now. The 2 sister chromatids are separating. Clear? So the centromere is splitting, that is breakup is happening, and 2 different chromatids are separating out. 2 sister chromatids are separating out one by one. That is, can you see here in the diagram? 2 sister chromatids are separated out, which was looking like this. Initially it was like this, right? In the previous metaphase we learned that it is not going back. Okay, I will change here. In metaphase we learned it was like this. Now this sister chromatid is moving this side, this sister chromatid is moving to the opposite pole. Opposite poles they are going. Okay, opposite poles. Now let me tell you a few points here. it is technically the shortest phase. It is the shortest phase. Here what is happening? Split of centromere is happening. Split of centromere. Splitting of centromere. Okay. Now, if the split of centromere is happening, what happens? If centromere is splitting, there is separation of chromatids. There is separation of chromatids. Very important, very important. Many questions will come. Many questions will come. They'll ask you at which stage in the cell cycle, the splitting of centromere is happening and the two sister chromatids are separated out. Separation of sister chromatids. Sister chromatid is happening. Is this point clear to all of you? Tell me. Sir, what is the difference between mitotic anaphase and meiotic anaphase? See, tell me what is happening right now here. In the case of your mitotic anaphase, in mitosis anaphase, there is separation of what? Separation of sister chromatids. Separation of sister chromatids in the mitosis. Separation of sister chromatids at anaphase. But in the case of meiosis 1, anaphase 1, in anaphase 1, there is separation of homologous chromosomes, which we will discuss in meiosis. Separation of homologous chromosomes in anaphase 1, which we will discuss in meiosis. But right now, mitosis. Let's stick to mitosis. Okay. If this point is clear to you, can I tell you the hidden point which I am trying to tell you? Can I tell you the hidden point which will help you to understand, the DNA content entire problems? Listen to me very carefully now. Now, can I tell these right here are your daughter chromatids? They are the daughter chromatids. Yes, they are the daughter chromatids. Now students, these daughter chromatids will act like daughter chromosomes. They will act like daughter chromosomes in the future daughter nuclei. The most important line of your anaphase, the most important part of your anaphase. That is, these daughter chromatids now, chromatids are separated out. These daughter chromatids will act like daughter chromosomes in the future daughter nuclei. Understand this. I'll give you a quick example. Tell me quickly in the chat. How many chromosomes are there in the entire cell? In a diploid cell, how many chromosomes are there? There are in total of 46 chromosomes. Yes, 46 chromosomes. And each chromosome has how many chromatids? There are 46 chromosomes. Each chromosome has 2 chromatids. We just now learned, right? Each chromosome has 2 chromatids. So in total there are 92 chromatids. 92 chromatids, right? Into 2. Do into 2, right? 46, doubling it. 92 chromatids. Okay, 92 chromatids are there totally. 92 chromatids. Now students, I just now told you, in this particular anaphase, how many chromatids are there? Tell me now. Obviously, sir, there are 92 chromatids here. There are 92 chromatids in this cell also. And I'm telling you students, each daughter chromatid is acting like a chromosome now. Each daughter chromatid in the anaphase. In anaphase, each daughter chromatid is acting like a chromosome. So how many chromosomes are there totally? If there are 92 chromatids here, can I also tell there are 92 chromosomes temporarily? 92 chromosomes temporarily. That is 46 chromosomes on this side, 46 chromosomes on this side. Understand. Temporarily, shortest phase, temporarily, there are 46 chromosomes on this side, 46 chromosomes on this side. Are you understanding? Because there are totally 92 chromatids here. 92 chromatids. 92 chromatids are separated out. 92 chromatids are being separated out. And each chromatid is acting like a chromosome now. Each chromatid is acting like a chromosome. So technically, 92 chromatids means 92 chromosomes. 92 chromosomes, students. 92 chromosomes are here. Now, are these 92 chromosomes single chromatid or two chromatid? Each 96 chromosomes are right. This 46 chromosomes on this side with one chromatid. With only one chromatid. So 46 chromosomes on this side with a single chromatid. 46 chromosomes on this side with a single chromatid. And that is the foundation of your 4 and 4C, which I will tell you in the next few minutes. Did all of you understand this concept? Tell me yes or no in the chat quickly. In anaphase, each chromatid is acting like a chromosome. So 92 chromatids means 92 chromosomes. 46 chromosomes this side, 46 chromosomes on the other side. This is the concept which I want my students to understand. Concept. Now. This here, we will stop here. Now, let me add one more point. To your anaphase. Students, this is also the best stage. It is the best stage to study. Best stage to observe the chromosome shape. Best stage to observe chromosome shape. That could be your V shape, L shape, J shape, or I shape. Metacentric, submetacentric, telocentric, acrocentric. All those shapes of chromosome can be studied best at anaphase. Clear? Now next thing is going to be see anaphase. What happening? Separation of sister chromatids. The separation of sister chromatids is happening. Sister chromatids are being pulled apart. Now, the last stage of your cell cycle. Last stage, students. This is technically your last stage. Last stage is called as telostage. Telophase. In telophase, what is happening? Listen to me very carefully. Telophase. Now, in telophase, what happens is the nuclear envelope is appearing again. Here also. Nuclear envelope is appearing. Okay, nuclear envelope is appearing. Let me divide the cell into two parts now. Now, chromosomes are losing its identity. And remember, students, your telophase is actually the opposite of prophase. Whatever happened in prophase, it is going back to prophase now. It is the complete opposite of prophase. Now, what is happening here? Chromosomes cluster at the opposite poles. Can you see? Chromosomes are clustering at the opposite poles now. Yes, opposite poles. Yes. And they're losing their individuality. Loss of identity. Can I write identity? Loss of identity, that is, till this stage you are able to see proper chromosomes. Now chromosomes are again decondensing. So they're losing the identity. They're losing their identity now. Okay. Your nuclear envelope is coming back. Nuclear envelope is reappearing. repairing. Your Golgi complex, endoplasmic reticulum, mitochondria, as well as your nucleosome, nucleolus, everything is coming back. Everything is coming back. Yes, everything is coming back. Okay. This is your telophase. Now tell me, in this telophase, can I tell, here we have 46 chromosomes. Here we have 46 chromosomes, this side. 46 chromosomes. 46 chromosomes this side, 46 chromosomes on this side. With single chromatid, one chromatid. Here also one chromatid. 46 chromosomes. Done. Any doubt till here? Ask me quickly. Any doubt here? Ask me quickly. Done. Because the next thing is going to be very important. See, telophase, the complete chromosomes will cluster towards the opposite pole. They're clustering towards the opposite pole. Okay. This is your anaphase. They're being pulled. By telophase, they're clustering. Nuclear envelope is coming back. All the different types of your cell organelles are reappearing. Now, students, the most important thing which you're waiting for till now, that is going to be the DNA content. That is going to be your DNA content. Okay. Now, can we start with the DNA content? Tell me yes or no in the chat quickly. Green us in the chat if I can start the DNA content, and you will be able to solve all the questions with the help of DNA content. All the questions now of mitosis. This is called as decoding biology. Students, all of these notes will be available. All of these notes will be available on my Telegram channel, Basavaraj Sir Biology, and also on Vedantu NEET English Telegram channel. All the notes will be available. Okay, don't worry. Now look at the DNA content. First, let's start with G1 phase. G1 starting. This was your chromosomes. This is your chromosome, and it is 2N, 2C. This is the N is the ploidy, C is the DNA content. Okay. Now what about S? In S stage, there is duplication. There is a doubling of DNA content. But is there increasing chromosome number? No. Chromosome number will remain 2N. 46 chromosomes will remain the same. But the DNA content will become 2C to 4C. Increase in chromatid. Okay, increasing chromatid. Now what about G2 phase? What happens in G2? In G2, nothing. It remains the same. 2N, 4C. Now the cell is entering prophase. Now this is your interphase, right? This is your interphase. Now onwards it is M phase. Now here. M phase. Now what happens in your prophase? Prophase, nothing is happening. It is 2N, 4C. This is your chromosome. Then we have metaphase. Metaphase, nothing is happening. 2N, 4C. Now. The most important part. Let me erase this. We need more space for writing. The most important part is going to be your anaphase. What happens in your anaphase now? Now students, can we go back to anaphase and just check once? Where is your anaphase? Telophase, anaphase, here. In anaphase, listen to me very carefully. What is N? N is going to be number of chromosomes, right? N is going to be number of chromosomes, N. In anaphase, what is N here? N is 23. 2N is going to be what? 2N is going to be 46. 3N is going to be 69. 4N is going to be 92. 4N is going to be 92. Now tell me, in the anaphase stage, how many chromosomes are there? Tell me, in anaphase stage right now, totally how many chromosomes are there? 46 chromosomes on this side, 46 chromosomes on this side. 46 plus 46, how much? 46 plus 46, how much? 92 chromosomes. 92 chromosomes. 92 chromosomes. And if there is 92 chromosomes, how do you write it? How do you write it? 4N 4C. See, DNA content there is no change. DNA content there is no change. But the chromosome numbers are 92 right now. Because each chromatid is acting like a chromosome. So 4N 4C. 4N 4C. Done. Now last one is your telophase. Last one is your telophase. Now telophase, I'll write it in two parts. One is the entire cell, and then I'll write per nuclei also, per nuclei. Two types I'll write down. In telophase, tell me in the chat now. In telophase, if you look at the entire cell, what is the number of chromosomes? Tell me, the entire cell. Entire cell, we have again 92 chromosomes, that's going to be 4N. But can I tell this part right here is just 2N 2C? This part right here is also 2N 2C. 2N 2C. Technically, your anaphase which was 4N 4C is being divided into two parts. The anaphase which was 4N 4C, 2N 2C is going this side, 2N 2C is going this side. Right. Are you understanding? Your 4N 4C, that is 2N 2C is going this side and 2N 2C is coming this side. That is per nuclei. Right. Understand the concept. So if I take it as per cell, if you look at the entire cell, it is still going to be 4N 4C. The entire cell is going to remain as 4N 4C. But if you look at per nuclei, it is going to be 2N 2C. 2N 2C. Per nuclei, 2N 2C. Now students, how did we start the cell cycle? The cell cycle started with 2N 2C. How it is ending? Ending with 2N 2C, but single chromatid, right? Here also we are single chromatid because in anaphase there is separation of chromatid. So how did we start? We started the cell cycle with 2N 2C single chromatid. How are we ending? 2N 2C single chromatid, and that is why your mitosis is called as what? Mitosis is called as your equational division. It is called as equational division. Understanding why it's called equational division? Do you understand why it's called as equational division, students? Because the division is happening. We're getting two nuclei. Later they'll go undergo cytokinesis, two cells. But is the DNA content same in the end of the cycle? Yes. Is the chromosome number the same in the end of the cycle? Yes. This is the beauty of your mitosis. Called as equational division. Understanding? So this particular content, DNA content, the prophase, metaphase, anaphase, telophase, gives you an idea that how beautiful the cells divide. Right? So can we do the last part of today's class? After that we'll solve some questions. Okay, we'll solve some questions later on, but right now can we do the last part, that is cytokinesis. See, all of you know, till now here we are done. Chirokinesis is over. Prophase, metaphase, anaphase, telophase is over. This part is over. Now let's focus on cytokinesis. Till now, if you do not have any doubt, students, quickly smash that like button, and I want you to tell me in the comment box today, because I have noticed some of you do not comment. Pavitra, Neha, Madhumita, yes, Javi, all of you don't comment only. Tell me in the comment section, did you truly understand mitosis today? Because all of you, I know you would have mugged up. You would have mugged up the prophase, what happens, metaphase, what happens, telophase, what happens. Only when you do the DNA content, you will appreciate the entire cycle. Let me know in the comment section today. Because everyone should know, right, what we are trying to do here. Now, let's go to the last part, that is cytokinesis. Cytokinesis is very simple, students. Cytokinesis is very simple, yet it is very different. When it comes to plants and animals. Yet it is very different when it comes to plants and animals. When it comes to animals and plants, it is very different. Very, very different. Very, very different. Very different. So how is it different, sir? How is it different? How is it different? Let me tell you. Now, in the case of animal cell, this is your telophase cell. Okay, like, too big. This is your telophase cell. Okay. In telophase cell, we have one nuclei here and one nuclei here. Inside that we have the chromosomes. Yes, chromosomes are there. Condensation happening. Now what happens, listen to me very carefully. What happens, listen to me very carefully in the case of your animal cell. That is cytokinesis. Division of, what is happening? Division of cytoplasm. Division of cytoplasm. Okay. Now here what happens is, in this corners, there is accumulation of some type of actin microfilament. Okay. Now because of this microfilament, what happens? Because of this microfilament, how many of you know balloons? All of you know balloons, right? All of you know balloons. Take a balloon and squeeze it from two sides. Take a balloon and squeeze it from both the sides. What will happen? It will go inside. That is what is happening. Now what are we doing? We are squeezing from inside. So it will end up becoming like this, something like this. It will end up becoming something like this, like hourglass. One cell here, one cell here. This is called as furrow formation. This is called as what? Furrow formation. Furrow. Can you see? Furrow. You're trying to pinch it. You're trying to pinch it from this side, like two sides. Balloon. Take a balloon and try to pinch it from both sides. It's called as furrow formation. And in the end, you'll get what? In the end, you'll get two cells. In the end, you'll get two different cells with two different nuclei. Okay? Two different nuclei, and all the chromosomes will be on the inside. Okay? Now. This is called as, see, outside physics, a little bit of physics. From outside to inside, you're pinching. Outside and pinching. What is this force called as? Centripetal. Yes. So this type of division is called as centripetal. One second. Let me write it down. That green is not visible, I think. Centripetal. Okay. From furrow formation is happening. Now students, what about in the case of plants? In the case of plants, you cannot pinch it. You cannot pinch from outside. There is no furrow formation. Why? Because in the case of plants, there is a very rigid layer. There is a very rigid layer on the outside. That is called as your cell wall. Very rigid layer called as cell wall. Inside we have the plasma membrane, and inside that we have the nucleus. Okay, this is your cell. This is your plant cell. Now what happens in your plant cell? In the case of plant cell, in the middle, can you see? In the middle, not from outside. In the middle, there is a plate formation. The plate formation is because of something called as phragmoplast cells. They are basically derived from your Golgi complex. They are derived. This phragmoplast cells from the inside, they'll make something called as a plate formation will happen from the inside. Now this plate formation is very simple. This is called as cell plate formation. Cell plate formation is happening on the inside. Now cell plate formation has happened. Now from inside, the two cells will divide. From here like this. This will go. It will divide. Okay. That is called as centrifugal. It's called as centrifugal division. It is centrifugal in nature. Let me show you a simple thing. See here. In the case of your animal cell, what is happening? Actin filament is coming, that is a microfilament. Furrow formation is happening. Yes. And finally, the two cells are dividing. But in the case of your plant cell, there is something called as cell plate formation. Cell plate formation happens from the middle. From the middle it starts. From the middle it starts and it goes. And then the two cells will divide. The two cells will divide. This is called as, in the plant cell, the cytoplasm is dividing from the inside. Centrifugal. Centrifugal. But, okay? Here it is centripetal. Centripetal. Understood? Understood? That's all. It's very simple. Very, very simple cytokinesis. Very simple. You don't have to break your head over this. You don't have to break your head over this. Very simple. Okay. So in the middle it is cell plate is forming and it is dividing in the case of plant. In the case of animal, furrow formation is happening. That's all. Formation happening. Now students, I can ask you one thing. Now, chirokinesis is over. Okay. Chirokinesis is over. Chirokinesis is over. Okay. Now, what if after chirokinesis there is no cytokinesis? There is no cytokinesis. If there is no cytokinesis, what will you do? See, chirokinesis is over. Chirokinesis is over, but there is no cytokinesis. No cytokinesis. So what will happen? Obviously, if this is the cell, it will have one nuclei here, one nuclei here. Eventually, it will divide again. More nuclei will be arranged inside. More nuclei will be arranged inside because the cytoplasm is not dividing. Only nucleus is dividing inside. So, can we get a multinucleated condition? Can we get a multinucleated condition now? Yes. This multinucleated condition is called as syncytium. It's called as syncytium. One second. Right here. It's called as your syncytium. Best example is your free nuclear endosperm. Free nuclear endosperm. That is nothing but your coconut water. Coconut water. What is this? This is a type of multinuclear condition. Multinucleated condition. Multinuclear condition. Okay? Multinuclear condition. Done. What are the significance we discussed in the starting? Reproduction, unicellular organisms, binary fission, same thing happening. And growth of development, repair and maintenance, growth in meristem in the case of plants, and also restoring nucleoplasmic ratio. Nucleoplasmic ratio is basically your DNA content as well as your cytoplasm. They are maintained. 2N2C starts with 2N2C, ends with 2N2C. Starting, ending, same to same. Let's see a question now. See this question now. All of you see this question. It's a NEET 2024 question. NEET 2024 question. Look at this question, all of you. Your time starts now. Your time starts now. Go. Answer in the chat. Answer in the chat. Let me look at the chat here. Students, quickly smash the like button, all of you. And I would want to share this video with everyone. Spindle fibers attached to the kinetochore of chromosomes during, didn't we just now learn? Metaphase. Metaphase. It is attaching to the kinetochore. NEET 2024 question. This year, students, this year. Look at this question. Which of the following never occurs during mitotic cell division? Which never occurs during mitosis? Mitosis. Coiling and condensation of chromatids is happening in your prophase. Spindle fiber attached to the kinetochore of the chromosomes attaching metaphase. Movement of centrioles towards the opposite poles, prophase. Pairing of homologous chromosomes, students. Pairing of homologous chromosomes. Does this happen? Tell me, does this happen anywhere? Pairing of homologous chromosomes, is it happening anywhere? Did we learn about anywhere? Did we learn homologous chromosomes are coming together? Anywhere did we learn? No. So this is your incorrect statement. Never happens. Never happens. 22 question. Select the incorrect statement with reference to mitosis. Splitting of centromere occurs at anaphase. Yes. All chromosomes lie at the equator in the metaphase. Yes. Spindle fibers attached to the centromere of chromosome. Tell me, do spindle fibers attach to the centromere of chromosome? No, they attach to the kinetochore. Right? They attach to kinetochore. Tell me, mitosis. What is the answer? Quickly tell me in the chat. Is the answer? Spindle fibers does not attach to the kinetochore, they attach to the kinetochore. They attach to the kinetochore. Chromosomes decondense at telophase. Yes, they condense at telophase. Done. Next question. Growth of a multicellular organism is due to mitosis. We learned that. Mitosis is also called as equational division and it offers genetic stability. Yes, that is why multicellular organisms are able to grow properly. It is providing genetic stability and it is equational division. Chromosome number and plasma is maintained. Answer is obviously C. A and R are correct. R is the correct explanation of A. It is explaining. That's why multicellular organisms are able to grow properly. Multicellular organisms are able to grow properly. Next question. With respect to metaphase, which of the following statement is incorrect? Incorrect statement. Incorrect statement. Incorrect. Incorrect statement, students. Okay. You might have been confused between these two. Remember, students, your mitosis is because of equational division and genetic stability only. Multicellular organisms are able to grow, right? That's why they're able to grow properly. Oh, I showed the answer. Okay. Complete disintegration of nuclear envelope takes place. Correct. Chromosomes become highly condensed. Yes. Metaphase chromosomes are made up of 4 sister chromatids. No. They're made up of how many? They're made up of 2 sister chromatids. All of you remember, right? Metaphasic plate. We have 2 sister chromatids. One sister chromatid, one more sister chromatid. So at metaphase, we only have 1 sister chromatids, not 2 sister chromatids. Easy. Now, anaphase promoting complex APX is a protein degrading machinery necessary for proper mitosis of animal cell. Okay. If APC is defective in humans, which the following is expected to occur? If all they are trying to tell you is what will happen if anaphase is not happening. That's all. If anaphase does not happen, what will happen? Chromosome will not condense, it will still condense. Chromosome will be fragmented, no. Chromosomes will not be fragmented. Chromosomes will not segregate, yes. Tell me, if anaphase is not happening properly, will the chromosome segregate out? Will the chromosome segregate out if anaphase is not happening? Obviously not. Chromosomes will not segregate out if anaphase is not happening properly. So 3 is the answer. 3 is the answer. Look at this question now. A stage of mitosis is shown diagram. Which stage is it and what are the characteristics? Obviously it is the metaphase. So these two are gone. Metaphase, spindle fibers attached to the kinetochore. Centromere split and chromatid separate. No, this is not the case because your centromere split and chromatid separate is in anaphase, not in metaphase. So what is metaphase? Chromosome move towards the spindle equator. Move to the spindle equator. Chromosomes are made of 2 sister chromatids. Yes, 2 is the answer. 2 is the answer then. It cannot be A because chromosome split and chromosome separate is a part of your anaphase. It is part of your anaphase. So 2 is the answer. 2 is the answer. Now students, this is a homework question. I will check. I'm telling you, I will check. Homework question. I will check the comments. I want you to comment. I want you to comment down in this particular answer in the comments, not here. In the comments, I will check. Sri Harsha, Madhumati, Savita, Harini, Hema, Naina, Ravindrani, Jai. I will check all of you. I will check in the comments. Homework question in the comment box. Also tell me if you understood mitosis today. If you understood mitosis properly, entire structure and everything, how the DNA content happens and everything. Okay. So let me know in the comment section. Let me know in the comment section. And where do you get the notes? All the notes will be available on Telegram channel, that is Basavaraj Sir Biology, and also on Vedantu NEET English Telegram channel. All the notes will be available in these 2 places. These 2 places, this is my personal Telegram. This is our Vedantu, amazing Vedantu Telegram channel. Notes will be available here. Where else? You don't have to go anywhere. You do not have to go anywhere, students. Everything is available at one spot itself. And if you want to solve more questions, how do you go? Students, if you go to the description of today's video, I'll show you. First of all, I'll tell you what you want to do. First of all, you want to do two things. If you go to description, so all our names are here. If you want to online test, if you want to start the test series from now only, you do not want to wait until the very end. If you just click here, you will get the one amazing test series which we have launched at Vedantu at just 1,000 rupees, year-long test series, not one day, 2 day. It is a year-long test series, You will get many tests, so if you want to solve more questions and everything, you should join a test series from now onwards. Do not wait until the very end thinking that, yes sir, I will do mock test later on. Do your mock test now. Do not repeat the same mistake. And students, if you go to the channel right now, and go to live, what is happening tomorrow? Tomorrow something special is happening. Something special is happening. Can you see this? Human reproduction. So tomorrow, 13th, right? Tomorrow 13th. 13th, I'll be doing your entire human reproduction. So I want everyone to go right now only and smash the like button, human reproduction, because we'll be doing the entire NCERT decode, every single thing in your NCERT. I'll make sure you understand every line, every concept from my side. So with that being said, thank you so much, students, and I will see you in my next class. Let me know in the comment section how was today's class, and smash the like button. And if you're new, subscribe to the channel. Bye bye. Take care. Have a great day, all of you. Have a great day. Yes, and this will be my zoology lecture. So if you have never seen me teaching zoology, this is your only and final chance to learn zoology from me. I'll see you. My only chance to learn zoology from me.